Hello friends, Mr. Ryan again. Today we're going to talk about active transport. We just learned about passive transport, how things get in and out of the cell without using energy. Today we're going to learn about how things get in and out of the cell that requires energy, and that's called active transport. Cells sometimes move materials against a concentration difference. What does that mean? Well, we talked about diffusion. We go from a high concentration to a low concentration. You're on top of the hill. To go to the bottom of the hill doesn't require much energy. But if you're at the bottom of the hill, you want to go to the top of the hill, that requires energy. The movement of materials against a concentration difference is known as active transport, and active transport requires energy. The active transport of small molecules or ions across the cell membrane is generally carried out by transport proteins, or protein pumps that are found in the membrane itself. So if we remember, we talked about the cell membrane, we talked about the phospholipids, remember phosphate, lipids, and we talked about proteins that are being embedded into the membrane. And when we talked about passive transport, we talked about proteins being used to help things get into the cell without using energy. Now with active transport, we have proteins inside the cell membrane that are protein pumps, and they require energy to move things in and out of the cell. Larger molecules and clumps of material can also be actively transported across the cell membrane by processes known as endocytosis and exocytosis. The transport of these larger materials sometimes involve changes in the shape of the cell membrane. A considerable portion of the energy used by cells in their daily activities is devoted to providing the energy to keep this form of active transport working. So when we're talking about the cells making energy, we're talking about the mitochondria, don't forget. So a lot of the energy made by the cell is used for active transport. The use of energy in these systems enables cells to concentrate substance in particular locations, even when the force of diffusion might tend to move these substances in the opposite direction. So now we're going to talk about endocytosis, right? We have endo and cyto. Well, cyto we know means cells. Endo sounds like in, take things in, okay? Endocytosis is the process of taking materials into the cell by means of infolding or pockets of the cell membrane. So if you look over here, we have the box on the left-hand side, endocytosis. But really what we're going to be looking at is over here on this right-hand part of the cell. So we have the particle, and it wants to get into the cell. The membrane forms a pocket around the particle. So the particle comes to the cell, a pocket's formed. The pocket then breaks loose from the outer portion of the cell membrane and forms a vesicle within the cytoplasm and goes inside the cell. Okay, large molecules, clumps of food, can even whole cells be taken up by endocytosis. And there's two examples of endocytosis are phagocytosis and pinocytosis. In phagocytosis, the extensions of cytoplasm surround a particle, package it within a food vacuole, then the cell engulfs it. So think of this as the particle, and this is the cell, okay? And the cell sees the particle, and it just engulfs it and surrounds it, sort of like Pac-Man, if you've ever seen the Pac-Man game. Okay, that's called phagocytosis. And amoebas use this method for taking food. Engulfing material in this way requires a considerable amount of energy and therefore is a form of active transport. In pinocytosis, cells take up liquids from a surrounding environment by forming tiny pockets along the cell membrane. The pockets fill with liquids and pinch off to form vacuoles within the cell. So with pinocytosis, the cells are just taking in liquid. Exocytosis, well, we know cyto means cells, exo, like exit, to get out. Many cells also release large amounts of material from the cell known as exocytosis. Again, the box is on the right-hand side, but what we're going to be looking at is on the left-hand side. So, the membrane of a vesicle surrounds the material, so we have the vesicle coming to the cell membrane, then it fuses with the cell membrane, and then the contents are forced out of the cell. During exocytosis, the membrane of the vacuole surrounds the material, fuses with the cell membrane, forcing the contents out of the cell. Friends, we are now done with active transport. So you've learned about the cell membrane, passive transport, osmotic pressure, and now active transport. As usual, have fun and enjoy learning.